However, before we talk about muscle physiology, it's important that we review a little bit of the anatomy. So, recall that you've learned that when our central nervous system makes a decision to send a signal to a muscle, it travels down a neuron. So, we have our neuron here. And this would be the end of the neuron, which, remember, is called the axon. So there is an electrical impulse, which, recall, is called an action potential. And an action potential forms when sodium goes into the cell, and then when potassium comes back out of the cell. And as this occurs, the electrical impulse travels down to the end of the axon. So this part right here is called the axon terminal. And in the axon terminal, there are small synaptic vesicles which have a neurotransmitter in them called acetylcholine. So this chemical called acetylcholine, it's going to be responsible for actually causing the muscle to become stimulated. So this little space right here that I'm drawing, first of all, this curved area is what is called the sarcolemma. And the sarcolemma is a very important word for you to know. It's the end of the muscle tissue itself. And the reason it's important is because it has little receptors on it, which are also proteins, called acetylcholine receptors. So this acetylcholine is released. It then binds to receptors on the sarcolemma. And an additional thing to know is this part of the sarcolemma, we call it the motor end plate because it's the part of the sarcolemma with acetylcholine receptors. So once the acetylcholine binds to their receptor, then the amazing thing that happens is there's another action potential that occurs. So it's almost like this up here is action potential number 1, and this would be action potential number 2. So really, we're combining several different things here. The first event up here at the motor neuron, and we call it specifically a motor neuron because it's a neuron that's associated with stimulating the muscle. Remember, the word motor is referring to muscle. So this first part is what's called the neuromuscular junction. And the junction is just what it sounds like. It's between the nerve and the muscle. That's where the acetylcholine binds to receptors. The next thing that we're going to look at next is what's called excitation-contraction coupling. And I know that sounds like a lot, but what happens is the muscle is excited. So that's when you see these arrows. So that means that this is action potential number 2, and the muscle is getting excited. This action potential is going to continue into the inside of the muscle, which is called the T-tubule. And I think it helps to draw this way because it's shaped like a T. So we have this T shaped right here. T stands for transverse tubules. So now the next thing that happens is the contraction. We had the excitation first. Now we're going to have the contraction. So this muscle is going to contract. And it does so because there are what are called sarcoplasmic reticulum. And back in the cell chapter, you learned that there was an endoplasmic reticulum within the cell. In muscle, we call it the sarcoplasmic reticulum because it's a little more specialized and it stores calcium ions. As you learned a long time ago, calcium is super, super important for muscle contraction. So this calcium, what's going to happen now, once excitation happens, then the calcium is going to exit the sarcoplasmic reticulum, and it's going to bind to proteins within the muscle. So you've seen the neuromuscular junction here. We have excitation-contraction coupling here. So I'm going to erase this part, and we're going to look at the third step, which is what is referred to as the cross-bridge cycle. And you learned a long time ago that when you hold your hands up to your face, you essentially are showing a cross bridge. So if you hold your hands up just like I'm doing it right in front of my face, I'd like you to notice that my left hand in this case is going to be 1/2 of a sarcomere, like that. The other hand, my right hand, is going to be the opposite side of the sarcomere. And so what happens now is this would be your hand, and it's what are called the actin filaments. So the actin is the small protein, and we call it the thin filament. So we are on step number 3 here, which is cross-bridge formation. So when a sarcomere contracts, the sarcomere, it's a super important part to know. It is the functional unit of the muscle, specifically of the skeletal muscle, which is what we're talking about now. So we have the thin filament, which is actin. That would be your fingers. The part that you really can't kind of demonstrate with your fingers is the thick filament, which is in here. And this thick filament is what's called myosin. And that myosin is going to form a cross bridge with the actin. So remember in the excitation-contraction coupling, we saw the calcium leaving the sarcoplasmic reticulum. That calcium then binds to proteins that are on the actin. Those proteins are called troponin. Now, if you work in the hospital or a medical community, you may have heard the word troponin levels being checked after somebody might have a potential heart attack. That's because the troponin molecule should stay in the muscle, the cardiac muscle in the case of a heart attack. So if it's released into the blood, that's a bad sign that maybe there's been muscle damage. So what happens now is the calcium binds to the troponin. And there's another protein that's kind of connected to the troponin called tropomyosin. What happens is it swivels out of the way. The myosin, the thick filament, can grab the actin, and when it does this, it pulls it inward. So if this is our resting sarcomere, when our fingers go towards each other, remember that's the actin, the thin filament, then we have a sarcomere that contracts. And that H zone, the middle part of the sarcomere, starts to disappear. So this is now called the cross-bridge cycle. And that's a big part of the muscle physiology that we need to know for this chapter. See you in the next video.